Okay, so let's begin with the session, and by the time other students will also join. So are my slides visible and am I audible? Just please confirm. Okay, thank you. Yeah, so this is your third week, and today we'll be talking about all the week 3 topics in which you have looked into proteomics and how the samples are prepared, and about protein quantification, and mostly the methods are related to mass spec and how different techniques are opted, and then how mass spectrometry is performed and what are different things. So along with this, we'll also practice few MCQs. We'll talk about the current literature, and I'll take your doubts if you have any regarding the assignment or any other doubts. So let's start. So firstly, this week was entirely dedicated to the proteomics topics. So what proteomics mean? So in this slide, I am showing you a paper which was published in 2021. It's about proteomics concepts and its applications in human medicine. So in this paper, they have very properly defined what is proteomics and what currently is ongoing and what are the applications. So when we say proteomics, first of all, it is made up of two words, proteo and omics. Proteo means it's about the protein content in the cell, and omics means study. So when we study the entire protein content, then it is referred to as proteomics. It is the complete evaluation of the function and structure of the protein to understand the nature of the organism, and mostly different techniques are used. So one such technique is mass spectrometry. It is an essential tool which helps us to analyze the proteins. So it is one of the measures to study the proteomics. And however, the biomarker discovery, which is important to identify the biomarker so that we can treat different disease. So for that, it is a major challenge with the proteomics, because what is proteomics is that it is quite complex and it is dynamic. So due to which, if we look at the genome, it is constant. But when we talk about proteins, it's complex and dynamic. So there are some challenges which one has to face while dealing with the proteomics. So therefore, we need to combine proteomic approaches along with genomics. That's why proteogenomics came up. So we cannot deal with the single technique only. We have to integrate different techniques to get better out of them. So in this paper only, they have also defined that when we talk about DNA, then that is at the level of genome. As we move to RNA, it becomes the transcriptome level, that which RNAs are expressed. So transcriptome will contain all the RNAs, mRNAs, tRNAs, rRNAs, and all other RNAs. When mRNA will code for the protein, that will be referred to as a proteome. And this proteome is diverse, because we have discussed in the previous lectures also that a single gene can encode multiple RNAs, and those RNAs can form different proteins. So single gene can have multiple protein expressions. So due to which this proteome is quite complex and it's dynamic, because some genes express certain proteins at some developmental stage. It will not express all its proteins at the same time. So due to which there is difference at the level of spatiotemporal, in which cell it will be expressed, or in which tissue protein will be expressed, and at what time the protein will be expressed. So these all things are quite dynamic. That's why the studying proteomics is quite challenging, because from where you are collecting the sample, that will determine that which proteins you will find. But if we talk about genome, then any cell you take, and you can take DNA from any tissue, it will be constant. But the concept is not same for the proteomics. So due to which it is dynamic. And then after proteome, if we move to the metabolome level, which constitutes all the metabolites, so they are also diverse. And this proteins further, the study after the proteomics is the metabolomics, which will ultimately determine that what will be the phenotype, or what will be the function, how the organism will behave. So ultimate thing is the phenotype. From genotype, how the things move till the phenotype. So entire things are shown in this slide. Now let's look at how this dynamic nature has come up. So from let me show you. So if you look at this portion, it is static. When we are looking at the genome level, so everything is static, it's constant, it is fixed. In each and every cell, DNA is constant, and we know there are only 20,000 genes. So this is just an approximate number, 20 to 25,000 genes are present in human genome. And then comes transcription, where the DNA is converted to RNA. So in RNA, there are mechanisms such as alternate transcription start sites, which will kind of synthesize different RNAs from a single gene. So this is one kind of dynamical process which will come and which will disturb the static nature of the gene. So when gene will make the RNA, there will be different RNA products which will be synthesized. So that's why these 20K genes nearly synthesize about 200K transcripts. So how these transcripts are synthesized? Due to the alternative transcription start sites. So this is one of the process which brings about the dynamicity. And then after the transcripts are synthesized, then comes the RNA processing step, in which 5' capping will happen, alternative splicing, alternative polyadenylation and RNA editing can happen. So these are also some of the processes which can change different RNAs which are synthesized from a single gene. So these are also some of the processes which can bring the dynamicity to the nature of the RNAs. Then after these RNAs are formed, now the next step is to synthesize the protein, which is known as translation. So how different proteins can be synthesized? That is governed by the alternative start and stop codons. So if among different RNAs, if their start sites and stop sites are changed, then multiple proteins can be synthesized. So they are saying that from these 200K transcripts, there is a possibility that we can synthesize about 1 million of proteoforms. So proteoforms are, you can say, different forms of the protein which arises from the similar transcript. So that is the proteoforms. And we will discuss more about what does proteoform mean and why we are not calling them as proteins and why proteoforms. So we will discuss that also. Then the next step is the post-translational modifications. So when the proteins are synthesized, the process doesn't stop here. The next steps also comes up, which is the post-translational modifications. So even same proteins can also have different post-translational modifications. In different tissues, similar proteins can have different modifications due to which they can behave differently. So this is also one of the contributing factor towards the dynamic nature of the proteins. And ultimately the next step is the protein-protein interactions. So a protein can interact with different other partners in different environments. So it depends that which other proteins are present. So in that way the interactions and the nature of that protein will be different. So these all are the contributing factors to the dynamic nature of the proteins. So now let's look at what does proteoform means. So this is the gene, as we all know, and it will give rise to mRNA, which will be the single stranded copies. These mRNAs can form different transcripts, and we also refer transcripts as isoforms. So when we say a mRNA is giving rise to protein, then those proteins are referred to as isoforms, and at the level of mRNA those are referred to as transcripts. So why we call them as transcripts? Because a single gene which is synthesizing multiple RNAs, so those multiple RNAs are coming from single gene, that's why we call them as transcripts. That means these all mRNAs are the product of a single gene. So then we will move to the isoform level, which means that a single gene is giving rise to multiple protein products, so those are referred to as isoforms. Now on these isoforms, or we can say the proteins, there will be some post-translational modifications which can be phosphorylation, glycosylation or acetylation. So these post-translational modifications will change these proteins or isoforms. So then the proteins which we obtain are referred to as the proteoforms. So this is the meaning of proteoform, when a protein is modified by the post-translational modification, but still those proteins are arising from a single gene. So we refer to them as proteoforms. So these are the variant of genetically encoded proteins. That means they are encoded from a single gene, and each proteoform is defined by the full set of modifications it has, whether those modifications comes from alternative splicing or post-translational modifications or any other source. Thus identifying these proteoforms requires a single molecule analysis of the full length proteins, and these proteoforms can have altered structure, function, interactions and even solubility, and these are exponentially more potential there are exponentially more potential proteoforms than there are proteins. So these are few of the proteoforms as I've shown. Now let's move to the next. So I hope till now there are no doubts. Okay. So now let's talk about these two terms, which are genomics and proteomics. So genomics means study of genome and their functions. When we talk about proteomics, it deals with the studying of proteomes and their functions. In genomics, we deal with the genomic sequences, how the variants are present in the genomic sequences, whereas in proteomics it includes protein sequence mapping and it also includes the 3D structure modeling. So in genomic level we do not compare the DNA structures, but when we talk about proteomics we also take into account the structural comparisons of the proteins. So we do not just compare the protein sequences, but we can also compare their structures and we can see that what are the structural changes between these proteins. For example, if I am comparing the normal person and the diseased person and they both of them has some protein, they have same gene which is expressing different proteins. Now I want to see that what are the variations among their proteins which are synthesized. So for that purpose I can go for the structural comparisons and I can suggest that okay, this structural change might have impacted the function of the protein. So for example, in a normal person the functional protein has the some binding site, or we can say the site which could interact with other proteins, but in the cancer state that structure might have disturbed that site, due to which no longer binding could happen or no longer interaction could happen. So that could be one of the reason that why in the diseased phenotype the particular expression is seen. So for that we can go for the structural modeling, and we can also look for the protein protein interactions, because as I have talked earlier that in different cells or different tissues different proteins can interact. There could be different proteins which are present in one particular tissue, whereas in other tissues some other proteins are present. So protein interactions will vary among different tissues, whereas their genome is still constant. So for genomics we generally go for the Sanger sequencing or next generation sequencing methods, whereas if we talk about proteomics we go for the mass spectrometry for the different analysis. We can also go for the affinity proteomics and protein microarray methods. So we will discuss all these techniques one by one in the later slides. So when we talk about genomics, it indirectly gives us some suggestions about the physiological states. For example, we can compare two person, normal and diseased person, and we can look at their genome and we can go for the SNP analysis and we can get the information of the SNPs. But with that we cannot confidently say that that particular SNP is involved in the disease. So for the greater or direct interpretations we have to go for the proteomics. Genomics can give us indirect clues, but it is not that much confident, whereas if we talk about proteomics it's quite confident. It directly specifies the physiological states along with the spatiotemporal resolution. So spatiotemporal resolution means the spatio word specifies the spatial arrangement or spatial distribution of the protein. So spatial means in different tissues or different cells different proteins are expressed. So if we obtain the sample from the brain, then we will be able to find the proteins which are only expressed in brain. But there could be certain proteins which are expressed in lungs and not in brain. So in that way there is spatial difference between the proteins which are expressed. So that will be known as spatial resolution. And temporal means time. So time based resolution, which means that there are certain proteins which are expressed in adults, whereas some other proteins are expressed in infants. So by dealing with the proteomics we can also identify that differences, that which protein is expressed in the which tissue and at what time, developmental stage, or at what time that particular protein is expressed. So that's why proteomics gives us the broader picture as compared to genomics, and we have to integrate both of them to kind of get to a single conclusion, because as I've talked that proteomics is quite dynamic in nature. So there are some challenges which are associated. So now what are the key questions that proteomics can answer? So first of all it can help us to identify the proteins. So what we mean with the protein identification, it means that if we are comparing two different people, for example a normal person and a cancer patient, so we can identify that which of the proteins are expressed in the normal person and which are the proteins which are expressed in the diseased person. So that kind of expression is known as differential expression. So this is one of the question which proteomics can answer, that is the differential protein identification. The second question comes protein quantification. So we can also quantify the proteins, that at what proportion or at what rate the protein is synthesized. For example, in certain tissues certain proteins are expressed quite quickly in greater quantities, whereas in other tissues it might not be expressed that in that quantities. So that quantification can also be done with the help of proteomics. Then comes protein localization. So with the help of proteomics we can also identify the location of a particular protein, that in which tissue or at which component of the cell particular protein is expressed. Then comes post translation modifications. So we can also study the post-translational modifications with the help of proteomics. We can identify that what are the different groups which are attached to it, whether there are some phosphorylations or acetylations that are responsible for the protein activation or localization. So these all things can be compared by dealing with the proteomics. Then comes functional proteomics. So we can also study the function of different proteins. For example, there are multiple pathways in our body in which multiple proteins are present. So there are some proteins which are present in a single pathway, they will interact with one another. So if two proteins are interacting, so we can kind of uh, comment that what function they might perform, because if they are interacting, they might be involved in a single pathway or similar pathway. So in that way, functional proteomics can also help us to tell or define the function of proteins and which proteins are interacting. Then comes structural proteomics. So proteomics can help us to identify the structure of proteins, and by identifying the structures, we can kind of look for the sites where the drugs can bind, drugs can attach, and in that way we can design different drugs. So let me give you an example. For example, you have heard about the enzymes and substrates. So enzymes are the catalytic agents which speed up certain reactions. So they bind to a substrate and convert it into product, and in that way it catalyzes the process so that it can happen faster. Similarly, drugs are also kind of the molecules which bind with the proteins, and they have some effect on that particular protein, or you can say substrate. So to identify those binding sites, we need to find the structure of protein. So that can be identified with the help of proteomics, and we can design drugs because we can look for the sites where the drug can bind, and in certain cases there could be multiple sites. So by studying proteomics, we can identify the site which is best for the activity of the drug. Then comes protein-protein interactions. So with the help of proteomics, we can study those interactions, and it is all about the key questions which proteomics can answer. Now I'll give you overview of different proteomic techniques. But before moving further, if you have any doubt, you can ask. Okay. Yeah, we can kind of comment. If we look at the protein-protein interactions, then we have a hint that if two proteins are interacting, then it is most likely that those two proteins might be involved in a single pathway. So with the interactions also, we can figure out a function to some extent. I cannot say that we'll completely be able to write the function, because there are some proteins which are involved in multiple pathways, or which can have multiple interactions with multiple proteins in different pathways. But to some extent, we can kind of comment on the function, what could be the probable or possible function of a protein by looking at the protein-protein interactions. So is it okay, or do you have any further query? Okay, so if anyone have query, then they can put it in the chat. Yeah, so there are multiple methods to look, means it depends that what kind of function you want to identify. In certain scenarios, uh, people want to look that if uh, there are different techniques. First of all, in for example, in the yeast to hybrid system also, we look for the different protein-protein interactions. We look for the sites where the proteins in the DNA also, we look for the sites where the proteins bind, and then it enhances its translation. Or uh, there are different other methods which are available with which we can comment on the function of that protein. There are some knockdown studies, but those studies are mostly at the level of gene, not exactly at the level of protein. So most of the studies where the protein function is identified, they are based on looking at the genome, not just at the protein level. In protein level, people just go for the PPIs and quantification and taking different tissues and then checking. Means in proteomics, people kind of have the idea that what is the phenotype, and then they look for the samples, and then they compare, and then they comment that this protein is overexpressed, and the phenotype, this particular phenotype, this protein is expressed, so they relate that protein to that phenotype. So in that way, functions are means assigned. But most of the studies, previous studies where the protein functions were defined, they were defined at the level of genes, where what people used to do, they used to knock down certain genes and then compare the expression, compare different phenotypes, and then comment that this gene is involved in this particular function. But till now also, nobody is kind of directly assigning the function to a protein, because one gene can have multiple proteins, so it's difficult to assign the function to each and every protein of that gene. But overall, to a gene, function can be assigned. So that is one thing. Okay. Yeah, and as we'll move further in the slides, so you will get more clue that how different techniques are used to look into different things. Okay, so this is the overview of all the techniques and the workflow in proteomics. This is a journal overview. So for purification, if we have the proteins, for purification we go for the chromatography based techniques. To detect different proteins, we go for ELISA, Western blotting and protein microarrays. And don't worry, we will discuss them in later slides, each and every technique. For characterization, if we want to characterize multiple proteins, we can go for gel based approaches or mass spectrometry, and the application of mass spectrometry are quite huge and it is most widely used technique. If we want to analyze the protein sequences, we can go for Edman sequencing, which sequence the protein from C terminal. So these are few of the amino acids there is kind of degradation of amino acids from the C terminal end, which help us to sequence the protein. For quantification, we can go for different labeling methods and ultimately using mass spectrometry by looking at the relative intensities, because these are the tags which are applied. And for structural analysis, X-ray crystallography and NMR spectroscopy is used, similar in the case as in the case of DNA. In protein also, we go for the X-ray crystallography and NMR, which provide us the three-dimensional structure of a protein, and we can go for the comparisons. And these methods are used, for example, if you obtain the novel protein sequence and its structure is not already available. So these are the technique which you use. And nowadays the new method has also come up, which is based on machine learning and deep neural networks, which is AlphaFold, which can predict the structure. So that is also one of the technique, but these are the experimental techniques. Then we can divide these proteomic techniques into two categories. One is low throughput and one is high throughput. So low throughput means which can only analyze the smaller number of proteins at a time, and its cost is quite low because as it is only analyzing smaller number of proteins. So example of the low throughput techniques are Western blotting, which is used to detect specific proteins in a sample, and ELISA, which is based on the antibody based method, enzyme linked immunosorbent assay, which is generally based on the bindings. Then comes high throughput proteomic techniques. So these were the previous techniques, low throughput. But then with the advancement, high throughput methods have come up. So what is the advantage of high throughput? That it is capable of analyzing thousands of samples at the same time. And its cost is high because equipments are quite costly. If you want to go for the mass spectrometry, then the device cost is quite high. But once you obtain it, it's worth it because you can analyze multiple samples at the same time. And examples are mass spectrometry and liquid chromatography along with mass spectrometry. So these techniques are combined to separate proteins by liquid chromatography and further identification using the mass spectrometry. So these are two categories. In this I have compared among high throughput and low throughput methods. So this is also similar to my previous slide only. If you want to study in detail that what is the difference, you can go through this slide. I won't repeat it again. Now I have divided the proteomics into different categories based on what method we are using. So in gel based, if we are using the gel to study the proteomics, it is known as gel based proteomics, and it generally uses the gel electrophoresis to separate the proteins based on their size or charge. And there are two techniques in gel based. One is 2D page, which is two-dimensional polyacrylamide gel electrophoresis, and another technique is differential gel electrophoresis or difference in gel electrophoresis, which is DIGE. And we'll go through them in the later slides in detail, so don't worry. Then comes gel free methods, in which we do not have to separate the proteins on the gel. We directly cleave them and go for the liquid chromatography and mass spectrometry without their separation on the gel. So we do not have to go through the limitation of gel based separation. An example of gel free method is shotgun proteomics, which directly digests the proteins using some peptidases or enzymes. Then comes targeted proteomics. So as the name suggests, targeted, that means something is targeted. So in this case we deal with the specific protein sequences of our choice. So in this already the set is already defined. We do not go for entire protein sample. We just decide certain protein We select certain protein, and for that specific protein we perform the analysis. That is known as targeted proteomics. And the example is selected reaction monitoring, which is SRM. And there is also another which is multi reaction monitoring, MRM, which is the category under SRM only, which quantifies the target peptides by monitoring specific ion transitions and useful for the biomarker analysis. For example, if I already know that this particular protein is involved in the disease, so I do not have to look into other proteins, I will simply deal with that single protein. So that is targeted proteomics. Then comes functional proteomics, in which I deal with identifying the function of the proteins. So for that we can go for the co-immunoprecipitation studies or Yeast-2 hybrid systems. We will look into them in detail in the later slides. So it explores the protein-protein interactions to understand the cellular processes and identify the potential drug targets. Okay, so these are few of the methods. There are some other also. I am not keeping them here because, as you know, proteomics is quite huge and dynamic. So let's move further to gel-based methods. So in gel-based proteomics, this is the procedure that how it is done. So first of all, we have to prepare a sample to study the gel-based proteomics. So sample can be obtained from any biological cell or biological sample we can take, and we need some lysis buffer in which we can keep that sample. Now we have to quantify the protein, because in gel-based method we require some quantity or some concentration of protein which is must. So we cannot deal with the smaller quantity of protein. We require some quantity which has to be put in the gel. So we have to go for the quantification so that we can check that whether the concentration of protein is enough and can we proceed with it or not. So that is the second step. Then comes gel preparation. So as these techniques are based on gel, so we have to prepare the gel, and most of the times polyacrylamide gel is used. So there is agarose gel also, but most of the cases in this deal with the polyacrylamide gels because of the pore sizes and differences. So this gel is better, and it is gel is synthesized in appropriate concentrations. The gel is prepared depending on the type of protein which you want to flow through that gel, for which you want to do the electrophoresis. Then comes protein loading. Now you have prepared the gel, you have to load your protein into that gel so that you can start with the separation of the protein. So protein is loaded into the wells which are created on that gel, and now you ensure that there is proper volume and concentration of the protein in those wells. The next step is electrophoresis. So in electrophoresis, it is constituted of two words, electro and phoresis. That means in the presence of electric charge, phoresis means movement. If you study the movement of something in the presence of electric charge, that is known as electrophoresis, and in this case we will look for the movement of proteins. So now in the gel, we will keep that gel in a solution or electrolyte, in which we will connect the ends of that container in which the electrolyte and gel is present to a charge, and now charge will flow through that sample, and which will help the protein to move in the gel. So gel has some pores, protein is present in the gel, electric charge is flowing. Now that protein will move in the gel in the presence of electric field. So the separation will happen on the basis of the size. It depends that at which charge is flowing and what is the size of the proteins. So in that way the protein will get separated on the gel. Then comes staining step. So now after electrophoresis is completed, we want to see that what is the position of those protein samples. So now we go for the staining, which will give us the bands, and then we will image the gel. Now we want to capture the positions, so we have to perform imaging so that we can compare, and ultimately we will analyze the data. We will identify the protein, we will check for the protein spots, and for further analysis we can go for the mass spectrometry by obtaining that protein and performing its digestion and following other steps of mass spectrometry. And ultimately validation and quantification steps comes up, in which we will quantify the expressions and identify the proteins. So we already have the information that okay, this protein is of this length, so its band should come here. So there is one standard or control which we know, so due to which we are able to compare the bands in the gel. So that is one thing. So that comparison will help us to distinguish between multiple samples or multiple proteins which are running in the wells. So this is all about the gel based proteomics. Now we will look into the two techniques which comes under this. So first is 2D page, which is two-dimensional polyacrylamide gel electrophoresis. So first of all, let's see why it is called 2D. Why it is two-dimensional? Because we are using two dimensions. First dimension is we will separate a protein on the basis of its isoelectric point, or that step is known as isoelectric focusing. Okay, so firstly, in the first step, we will add the protein mixture here and we will provide the electric field. So in the presence of electric field, that protein sample, it will start its movement depending on the isoelectric point. So isoelectric point is the point wherever the complete charge on the protein is 0, when it is in the neutral state, when there is no charge on the protein, that is known as isoelectric point. So in the pH where that charge becomes null, that is known as isoelectric point. So firstly, the separation will happen on the basis of isoelectric point, and in the second dimension, after we have obtained the proteins sample on the basis of their isoelectric points, we will go for SDS page, in which these proteins will be separated by the molecular weights. So different molecular weights will be present. So different molecular weight proteins are present. So larger proteins, when they will move through the gel, they will take some time, whereas the proteins which are smaller in size, they will move quite quickly. So in that way, in this second dimension will give us the larger proteins towards the negative terminal and positive smaller proteins towards the positive terminal. So this positive means where we are also flowing the electric charge so that these things can move. So this will give you the idea. So in this way, it will separate the proteins firstly on the basis of the charge and secondly on the basis of the molecular weight. Okay, so in the later slide, I will tell you that how this isoelectric focusing works. So there is a concept of Zwitter ion. So I think you might have heard this word during your chemistry lectures. So Zwitter ion is an ion in which the overall charge is neutral. That means similar number of positive and negative ions are present. So that makes a particular amino acid constant. That means it doesn't look like a positively charged or negatively charged. The charge is balanced, so that is the Zwitter ion, that is equal number of NH3 positive and CO negative groups. So in this case, I am talking about the amino acids. So in that way, amino acids have the N terminal and C terminal. So when both of them is having the similar charge. Normally, if we look in the cell, so the protein have these amino acids, so they are not in their stable states. They are not like in this way. Glycine is linked to some other amino acids in the protein chain, but whenever they separates, they are not in their stable form, they are charged. So they can exist in the Zwitter ion form depending on the pH of the solution where they are present. So in our body, in different cells have different pH, different organelles present in the cell have different pH. So in that way, amino acids can have different charges, and isoelectric point is the pH at which these amino acids have overall 0 charge. So if we talk about amino acids, their isoelectric points can range from 2.8 to 10.8. So in this case, I am taking the example of glycine. So it is mentioned that glycine has its isoelectric point at 6. That means at pH 6, it is behaving as a Zwitter ion. That is, it is neutral. So it exists as a positively charged species at pH below 6. So if pH is less than 6, it is positively charged. So in the case of isoelectric focusing step, which is the first step, so in that case, what will happen whenever we will flow the electric charge? So as it's positively charged below 6, so it will try to move from below 6 to 6 so that it can become neutral. So in this way, different amino acids which are present in the protein will move towards their isoelectric points. So in the case of glycine, it will move towards pH of 6. Similarly, different other amino acids will have different isoelectric points. So in that way, they will move according on the basis of their isoelectric points. And for example, if pH is greater than 6, then this particular glycine will have a positive charge and it will donate its H positive and it will convert to negatively charged ion. And one more thing which I want to add is that the positively charged amino acid, they will move towards the negative electrode, because positive will move towards negative, positive will be attracted to the negative electrode, and negatively charged amino acid will move towards positive electrode. And one more thing which I want to add in this previous slide, so as we can see that more acidic proteins will be moving towards the positive terminals. So with this we can say that the proteins which are acidic in nature, they are negatively charged, whereas the proteins which are basic in nature, that means the proteins which contain more of the basic amino acids. And in basic amino acids, for example, lysine, histidine and asparagine, these are kind of positively charged amino acids which are basic in nature, so they will move towards negative terminal. So in this way, proteins are separated on the basis of the charge which they carry and what is their isoelectric point. So in that way the movement will be observed. Okay, so the next concept is dyes. So I hope till now everything is clear. Okay. So now let's look into dyes, which is differential gel electrophoresis, or some people call it as difference in gel electrophoresis. So dyes is an kind of advancement to the 2D gel electrophoresis or 2D page. So why it is advancement? Because it allows to identify multiple or simultaneously run multiple samples at the same time. So in this, what we use on an addition to 2D pages, we can use the fluorescent cyanin dyes. So in that way, multiple samples can be labeled with different fluorescent dyes, and in that way, in the single gel only, we can run multiple samples which are labeled with different fluorescent dyes, and in the end, with the help of the to identify those fluorescent signals, we can capture the fluorescent signals and we can distinguish between multiple samples. So that is one of the advantage, because if you perform 2D page for multiple samples separately, then there are some issues such as different gels are, because you prepared gels differently for different samples, so there could be some variations or fluctuations in the conditions that how the gel is prepared, what are the concentration of the ingredients. So due to that we are not much confident, but in this case dyes we can use single gel, so we do not have the issue, we do not have to think about that there might be some issue with our gel because we are running it in the single gel. So first step is to perform to attach the fluorescent cyanin dyes and then ultimately combine those different mixtures in the single place and put it and let it go for the 2D gel electrophoresis. So we'll perform similar procedures as we performed in 2D page, but only in addition to it we have labeled different samples differently. So ultimately similar thing will be performed here also, and ultimately we will obtain different bands and different uh, different bands will be obtained, and we can distinguish between those bands by looking at the fluorescent signal. So firstly they will be separated on the basis of their charge, and then they will be separated on the basis of their molecular weight. So this is one of the advantage of dyes, that we can look multiple samples in the single gel. And in this slide I have put a table of comparison between 2D pH and dyes. So first of all, in dyes, as we know, it performs separation of protein in 2 dimensions, isoelectric point and molecular weight, which is done by the SDS page. But dyes is advancement which use the fluorescent dyes to label different samples and then perform the simultaneous comparisons by using 2D page only. So the purpose of 2D page is to identify and compare the protein expression profiles across different conditions or different treatments, whereas in dyes it enhances the sensitivity and it reduces the gel to gel variations, because as I discussed earlier that in the 2D page we use different gels when we have different samples, but in this case we can simply go with the single gel for different samples. So we do not face the limitation that there could be some variation due to gel. So the technique is 2D page is simple, the 2-dimensional gel electrophoresis, whereas in dyes labeling followed by the 2-dimensional gel electrophoresis. So these are some applications such as protein profiling, post-translation modification analysis and differential expression studies, whereas in dyes we can go for the comparative proteomics, because we can have multiple samples, 3 to 5 samples also we can compare at the same time using dyes. So comparative proteomics can be done, biomarkers can be discovered, validation of protein expression differences can be done with the higher accuracy. So example of 2D pages, that if you want to compare the protein expression in healthy or diseased tissues, and in case of dyes we can compare the protein expression between the treated and untreated samples with improved quantitation. So this is also one of the advantage, because we can at the same point we can analyze multiple things. Now I'll move to the gel free proteomics. So till now we have looked into the gel based techniques, and now we are moving to gel free proteomics. So in gel free we do not have to prepare the gels. So in this, sample preparation will be similar. We will obtain the protein sample from the biological tissue or biological sample, and then we'll quantify the protein, because in this step also we need to have certain concentration of protein. So we'll ensure that concentration of protein in the sample so that there could be equal loading. Now the third step here is protein digestion. So in this we'll digest the protein, we'll fragment it, digest the protein into peptides. In this we use specific proteases which facilitate the analysis. Now we will separate this protein. So for the separation of that digested protein we'll use the liquid chromatography, and in this case for example we can use reverse phased liquid chromatography. Then we will go for mass spectrometry to analyze the separated peptides, and mass spectrometry will help us to identify the identity of that peptide, that what are the sequences in the peptide, and it can also help us to quantify them, that in what quantity those samples are present. Then next step is data acquisition. So these steps are quite general which will be followed in each and every technique. So now after performing mass spectrometry we will get the spectral, we will get the spectras, then we will acquire that spectra, we will collect that data. Now next thing is that we will analyze that spectra and we want to identify that which peptides are there. So next step is data processing. From the spectra we will process it, we will quantify it, then we will analyze it. What we can analyze is the protein expression levels, we can compare the interactions, and we can gain some functional insights from it, and ultimately we have to validate our findings to check, because there could be multiple spectra which could be obtained, so we have to cross verify that whether the spectra which I obtained and the peptide tagging which I have done. So what we do in mass spectrometry, we get the spectra and we compare its mass to charge ratio and we give some peptide sequence to it. So spectra is the signal, sequence is the what we give to that signal. So there are certain scenarios where that assignment could be wrong, there are some probability that we have assigned the wrong sequence, so we have to do that validation to be sure that the assignment which have been done are correct. So validation is very important. Now I'll give you the example of gel-free proteomics. So in this slide I have put the comparison between the gel-based approaches which we have seen and gel-free approaches. So sample preparation is same, we can obtain it from the biological sample, biological tissue, or we can also obtain the protein from the bacterial culture. We will have supernatant, we will perform the precipitation to obtain the protein, and ultimately protein will be extracted, its concentration is maintained. Then in gel based we can go for the SDS page, 2-dimensional electrophoresis and dyes. So in SDS page the separation will be on the basis of the molecular weight, whereas if we use 2D page or 2D electrophoresis, the separation will be on the basis of both the charge as well as molecular weight. And in dyes, charge, molecular weight and different samples can be multiplexed at the same time. So these all are used to separate the proteins on the multiple criterias. Then next step is to digest, because ultimately we are going to quantify it, so quantify and identify it, so which is done with the mass spectrometry. So gel based is the first step in the analysis. Then we go for tryptic digestion, we obtain the peptides, we analyze those peptides using mass spectrometry, we can separate those peptides with the liquid chromatography and then go for mass spectrometry, and then we can analyze the data. But if we talk about gel free approaches, so in this case we do not separate the protein on the basis of gel or by using gels, but we simply go for the tryptic digestion. So we use the tryptic enzymes, we use certain enzymes which will cleave those proteins into smaller peptides, and as we have obtained the peptides, we can simply go for the mass spectrometry. So this is peptide fractionation, in which we have multiple peptides, so we can put it into different fractions, and then we can go for mass spectrometry and ultimately analyze the data. So these processes will be similar which we have discussed previously, but the main difference is that in the case of gel free approaches we do not use gel, whereas in gel based approaches we use gel for separating the different proteins. Now the third thing which we discussed was targeted proteomics. So as we have discussed that in this case we emphasize on specific proteins or peptides. So we already know that which protein we want to study, so that's why it is kind of targeted proteomics. We do not care about all the proteins which are present in the sample, but we deal with only single protein. So the quantification of single protein. So first step is sample preparation, in which we obtain the sample, then we quantify it as we have discussed, and then the third step is peptide selection. So in that case we identify and select the specific peptides for analysis. So we know that our protein of interest could have these peptides, so we will specifically select those peptides. We can also synthesize the synthetic peptides to use as an internal standards, so that could be a standard or calibrant, so that we will know that which peptide to select if we have the list of the standards. So this is the peptide synthesis step. Then comes sample digestion. So as we have discussed that we need to digest the protein into different peptides so that we can proceed with the mass spectrometry. So for sample digestion, we will digest the protein into peptides using proteases. So proteases are the enzymes which target the protein and break it into smaller peptides. And then we will perform chromatography to separate those peptides, and for that liquid chromatography is mostly used in these scenarios. And then we will go for mass spectrometry, which will analyze the peptides on the basis of their mass to charge ratio. And in case of targeted proteomics, we go for SRM and MRM, so selected reaction monitoring or multiple reaction monitoring. So these are two techniques which comes under targeted proteomics to quantify specific targets, because as the name suggests, selected reaction monitoring. So we are selectively monitoring a single reaction. Then comes data acquisition. So after mass spectrometry, data is acquired, it is analyzed, and it is validated. So these are the common steps which have to be performed. Okay, so this is all about targeted proteomics, and in this I have given the example. So this is discovery proteomics and this is targeted proteomics. So when we talk about discovery proteomics, so in the sample there could be multiple proteins which are present. So I do not care about any protein, I am just performing the MS of all those proteins which are present in the sample. But when we talk about targeted proteomics, then I make sure that in the sample I will deal with only specific protein. So I will obtain that particular protein and then proceed with its digestion. So before the step after obtaining the sample, I have to perform the peptide selection step. So that is about the targeted proteomics, that I have to select one protein of my interest, or the proteins which are of my interest, so I have to select them first. So it's not like discovery proteomics. So in that way, the spectra which I will observe after the mass spectrometry will be specific to that single protein, whereas in case of discovery proteomics, it will perform the shotgun experiments of all the proteins which are present in the sample. And in the case of targeted proteomics, we call those techniques are as SRM and MRM, which is selected reaction monitoring and multiple reaction monitoring. So MRM is the category of selected reaction monitoring in which at the same time multiple peptide fragments are selected, and ultimately we can identify the relative and absolute quantifications of the proteins of interest. So this is just the name which is attached. Techniques remains the same, but it depends at what you are dealing with. So in the targeted, you are dealing with single protein, remaining all techniques are constant. For example, in everything we go for the mass spectrometry only in the end, and for the purification we go for the liquid chromatography. So those steps will remain same, only the on which thing we are emphasizing. So in that way the name are given. So in this case, targeted proteomics. Similarly comes functional proteomics. So when we are trying to study the function of a protein, we call it as a functional proteomics. So in this case also we obtain the sample. It depends from where we are obtaining. Then we go for the quantification. Now we have to perform the functional assays. So we design assays depending on what kind of analysis we want to do. We can go for identifying the protein-protein interactions, or we want to look for the enzyme activity. So enzymes are also the proteins. So if I want to look for the enzyme activity, then also I'm kind of dealing with proteins only. So I can go for looking at how well it is catalyzing some reaction, and we can also check that a single enzyme can have multiple substrates. So enzymes are quite specific. And then certain enzymes which are also referred to as promiscuous enzymes, which can have multiple substrates, or there are some proteins which can bind to multiple interaction partners. So we can identify that with which protein the interaction is better. Similar in the case of drugs. So a single protein, multiple drugs can interact with single protein and can have different effects. So which drug is better, I can also identify that with the help of functional proteomics by comparing different things, that what is the strength of its interaction, what is the concentration in which interaction is happening. Because we all know that when we talk about enzyme substrate reactions, we say that there is some competition between those substrates. So the substrate which is better, and it will compete with other substrate, and it will outperform, it will bind with the enzyme, and other substrate will not be able to bind, because that single substrate which is strong, that will bind. So in that way we can also identify the drugs which could be better to target certain proteins. So those assays can be performed in the functional proteomics. We can also look for the protein interaction studies, and for protein interaction studies we use co-immunoprecipitation. That means at the same time we immunoprecipitate two different proteins by using antibodies, or we can go for the Is2 hybrid system, and we'll discuss this also. Now for functional validation, we have to validate the functional role of proteins through perturbation experiments. So in perturbation experiments what we do is that we disturb some structural part of the protein to check that what phenotype it produces, now what is the influence of those changes. So with that we can kind of comment about the function, that if we disturb these things then these are the influence on the functionality of a protein. So in that way functional assignments can be done. We can validate the role of the protein. Similar other functional assays can be performed. Now ultimately data will be collected from those functional assays. So we will see that how this Is2 hybrid and co-immunoprecipitation works. So after that we will obtain the data, and these data can be in the form of interaction networks, or there could be some activity measurements, or we can also look into the strength of interaction between two different proteins. So that could be the data which we can obtain depending on that what we are performing, what analysis we performed. So after obtaining the data we will perform the analysis of that data, that what are the different things which we can observe. We will interpret those things, and after that we will go for the validation and cross checking. So in that way if we look at the applications, so there are broad applications but few of them are like drug discovery and disease modeling, and we can also compare between two different people, normal and patients, that what are the differences. So how the functionality of the protein changes among different phenotypes or in different conditions. So those things can be done in the functional proteomics. Now this is the example that what kind of things we can get from functional proteomics. So one thing is protein-protein interactions. So we can identify protein-protein interactions. We can also identify the localization of proteins. We can also look for the post-translational modifications. We can study the structure of protein. We can also look for the turnover. So turnover is that, for example, in case of enzymes, so turnover means how fast it is synthesizing some product. So that is the turnover number. Or if we talk about proteins, then how well they are translated, how more and more proteins are synthesized, that is known as turnover. So these all things can be studied under the functional proteomics to some extent. Now in the right side if you see, this is for example in this case I want to identify protein-protein interaction. So what we do is we have a protein bait. For example, I have a protein, I want to see that what are the interaction partners of this protein. So I will have this protein bait, I will attach it to the specific ligand. So there will be a specific ligand which is attached to a bead. Now this ligand will be specific to my protein, and now my protein will bind to this ligand, and now binding has happened. Now what I can do is I can give cellular extracts which are incubated with this immobilized bait. So now this bait is immobilized because I have bind it to the specific ligands. So now it is immobilized, and these are the cellular extracts which can contain different proteins. Now I want to check that which protein will bind to my bait. So in that way some proteins will bind whereas others will not bind. So in this case if you see, this particular protein is only binding. So this will bind to these immobilized protein baits, whereas the unbound proteins will be removed because I will perform the techniques, chromatography or any other technique to move out the unbound proteins. Now this binding has happened. So what I can do is I can further elute the bait and the partners, and after that I can perform SDS page to identify that which protein has bound to my protein of interest, and ultimately MS identification can be done. So in that way what we have done here is that we have identified the interacting partner of the protein bait. So protein bait is the protein which is the query protein for which we want to identify its interaction partners, and we have taken a cellular extract which contain different proteins. So ultimately what we have obtained that which protein in the cellular extract is most likely to interact with the protein bait. So in this way protein-protein interactions can be identified with the help of functional proteomics. So I hope till now everything is clear. Okay. Yeah. So then I'll proceed with my next slide. Okay, so in this I have given you the brief overview of all the techniques. So I'll just briefly go through them one by one. So first technique is two-dimensional gel electrophoresis, which is involved in the proteomic analysis, which we have discussed. Then comes Western blotting. So in Western blotting, this is also based on the gel-based separation of the proteins. So it also detects specific proteins by using electrophoresis, and it is followed by the transfer of the membrane and antibody-based detection. So ultimately, in the gel, whenever we load the protein of interest, then we have some probe, or we can say we have some specific antibodies, because we want to identify the protein of interest. So those antibodies will be specific to our protein of interest. So from that gel, we will transfer that gel, will transfer those proteins to a membrane, and then we will see that antibody will bind to only to the protein of our interest. So in Western blotting, we will be able to identify that whether protein of interest, or the protein which we want to identify, whether it is present or not. But the limitation is that because we are using antibodies or some specific agents which are kind of interacting with that particular protein, so it is not possible to find the antibodies for all the proteins. So in that case, Western blotting will be limited to specific proteins only. Only specific proteins will be identified, others could not be identified. Then comes mass spectrometry, which is widely used, and it is used to analyze the mass to charge ratios of ions to identify and quantify the proteins and the peptides. So peptides are the smaller parts of a protein. So when protein is broken down into smaller units of 20 to 25 amino acid long, then that are referred to as peptides. So these are the smaller parts of a bigger protein. So we can quantify them by using mass spectrometry. Then comes liquid chromatography, followed by mass spectrometry. So this is also one of the best technique, because separation is done by the liquid chromatography, and ultimately mass spectrometry is done for the quantification and identification. And another technique is the protein microarrays. So in protein microarrays also, like DNA chip or DNA microarrays, protein microarrays are also like the immobilized proteins which are attached to a solid surface, or the antibodies which will be specific to certain proteins. So when we provide our sample, then only the means protein present in the sample which are specific to those antibodies can only bind. Other proteins won't be bind because we'll wash it. So ultimately, what we obtain in the protein microarrays, we will find the protein of interest, that whether it is present in the sample or not. So similarly, this is also limited because we are limited by the antibodies which are specific. So it is difficult to find the specific antibodies for all such proteins. And if my protein is so large that it cannot bind to that surface, so then also I cannot go for the protein microarrays. So in that way, this approach is also limited. Then comes surface plasmon resonance. So it measures the real-time binding interactions by detecting changes in the refractive index. So in this case, whenever refractive index changes, then with the change in the refractive index, I'll be able to know the binding interactions, that whether interactions are strong or weak. So this is generally used for the protein-protein or protein-DNA interactions. Then quantitative proteomics. So it is also the kind of big category, that in the cases where I am looking for the quantity, that how in different samples what is the quantity of protein, or I am comparing the protein abundance, how it is different. So that is referred to as quantitative proteomics. So it is also divided into the label free and labeled techniques, so which we will study in the later slides. Then comes SILAC. So this is also kind of a labeling technique. So this is the labeling technique, and ultimately followed by mass spectrometry, because in this step samples are labeled by using stable isotopes in the cell cultures. And this is means these are some of the techniques which are the initial techniques, means these have to be followed up by the mass spectrometry for later on analysis. And then comes TMT, which are the tandem mass tags. So this is also chemical tagging method, in which the tagging is done for the multiple samples, and ultimately mass spectrometry is done to distinguish or to perform the relative quantitation or quality check of the proteins. So this is the overview of different proteomic techniques which are present, and there are many, so we will not go through all, but these are the techniques which are covered in your week 3. Yeah, so now there is one more thing. Protein microarrays were also discussed. So I have gone through this paper in which they have very clearly defined the protein microarrays as a tool for the functional proteomics. that how these microarrays help you, and they have very clearly explained that what are the different types of microarrays. So first of all, microarray is a high throughput technology in which what we have is we have a solid surface which could be glass, or it could also be some other solid surface in which the immobilized proteins will be coated, or antibodies could be coated. So these antibodies or these proteins, they have to be specific to the protein or to the sample which we want to analyze. So it has to be specific. And now what we can do is we can analyze the protein interactions, expression levels and functional activity across numerous samples, because in multiple samples multiple proteins are present, and I know that this antibody will bind to this particular protein. So among different samples I can, if there will be some binding happening, so I can comment that this sample contains the protein and this sample does not contain. So that comparison can be performed with the help of protein microarrays. So there are two categories of the microarray based analysis. One is abundance based microarrays and one is functional based microarrays. So when I am looking for the abundance, means if I am comparing the abundance, that what is the quantity of different proteins in different samples, so that is known as abundance based microarrays. Whereas if I am comparing the functionality among different protein samples, so that is known as functional based microarrays, where I collect the so in this what is done is it's a high throughput method by printing a collective of target proteins on the array surface and assessing their interactions and biochemical activities. So this is kind of where I am dealing with the functional level and not with the numbers or quantities. And also focus on this analyte specific reagents. So analyte is the thing which we are analyzing, and specific reagents means the reagents which are attaching to my analyte, so which are specific for my analyte, so we have to use them. Now I have, means in this paper they have given both these microarray techniques. So in the case of abundance based microarrays we have three techniques. One is direct labeling. We can also perform sandwich immunoassays and reverse phase protein blots. So these methods have certain limitations and certain advantages. So if we talk about direct labeling, in this direct labeling is done of those proteins, multiple proteins in the sample, we will directly label those samples. So what advantage this offer is that we can assay multiple samples simultaneously, because we are directly labeling multiple samples. So we can compare the two states with different color labels, because I am using different color labels to distinguish between the different samples. But the disadvantage of this technique is that it requires chemically modifying samples, so which could have some cross reactive analyte specific reagents which can give false readings. So there could be some cross reactivity which can happen. So there are chances of errors which can be generated by the direct labeling. So then comes sandwich immunoassay. So it avoids labeling the samples. In this I am not going to label my samples. So in that way the specificity is increased using the two analyte specific reagents, because in this case I am using two different analyte specific reagents, and the limitation is that I require two analyte specific reagents for each target, because in this case we are only using single ASR, but we are labeling samples differently. But in this case I am not labeling the samples, I am directly using two different ASRs for both the samples. So I am not disturbing my samples as it was disturbed in direct labeling, but I require two ASRs and it is more difficult to multiplex. So this is one of the limitation. And ultimately is the reverse phase protein blot, which is the rapid screen for many samples at the same time. But in this case also limitation is that there could be cross reactive ASRs. So the analyte specific reagents can cross react and give false readings, because it might appear that they are interacting and ultimately they are not interacting with one another, but actually my ASRs are cross reactive. And narrow dynamic range, and other limitation is that rare proteins may not be captured. So if proteins will be rarer, then for them interaction capturing is quite difficult. I cannot be able to capture their abundance. So we won't go into much more detail. This is just a brief overview of these three techniques. Now we'll move to the functional based microarrays. So in abundance we will deal with the, in two samples, what is the abundance of protein, whereas if I look into the functional based microarrays, so in this case we have to deal with the functions, that in different samples what are the interactions which are undergoing. So first technique is the chemical linkage. It is the random orientation, and in this we look for the covalent binding. So this is the in this case what will happen is the target protein will bind covalently to the surface, so in that way we will be able to know that whether interactions are strong or not. So it bound close to surface, that is one of the limitation, and second limitation is that chemical linkage may affect the folding, so which can create the error in analyzing the function of that protein. Then comes peptide fusion tags. So in this case we can tag different samples with different peptides, so in that way different samples are labeled differently with different peptides, so in that way there will be unique orientation and protein held away from the surface, so protein are not closely held to the surface. But the limitation is that because we are using these tags, so their N and C terminus might means always get blocked, and proteins must be tagged. So in every case we have to tag, so it's very difficult to find so many peptide fusion tags, so that's why that is one of the limitation. But it's still better than the chemical linkage because it's holding proteins away from the surface. Then the third method is the self-assembling protein arrays, in which the proteins are produced just in time for the assay, and proteins are expressed in mammalian milieu like proteins are expressed in the normal cells. Similarly, the expression can be observed here as well, and there is no need to express and purify the proteins, and printed arrays are very stable. But the problem is that same as peptide fusion tag, that we have to tag multiple proteins, so that is one of the limitation. So this is all about the functional based microarrays. You don't have to go into the details of the techniques, you just have to remember the names, that what are the different techniques which are used for the functional based microarrays. That is all about them. And I think the question related to abundance based microarray was also in your assignment, so I hope this will help you to solve that question. Yeah, now I will talk about the surface plasmon resonance. So before moving to this, I'll again ask that is there any doubt till now? Okay, thank you. And don't worry about the previous technique, because there are many techniques which I am going in detail, but you don't have to remember them in that much detail. You just need to get the idea, you have to clear the basics, that what is the basic thing behind them. So now I'll discuss about the surface plasmon resonance. So as we previously discussed, that this is for the protein protein interaction analysis, and what happens is there is change in the refractive index. So refractive index, so it is a optical technique, because we are talking about the change in the how the refractive index changes in different mediums. So it is an optical technique, and it helps to measure the molecular interactions in the real time. So in real time means at the same time you are getting the signals, so which is known as real time interactions, and it involves directing the plane polarized light at a metal film under total internal reflection conditions. So SPR signals, which are the surface plasmon resonance signals, is sensitive to change in refractive index of the medium on the sensor chip. So here you can see this is the chip, and these are the immobilized ligand samples which are immobilized to this chip. So when we will pass polarized light through this surface, so if there is any change, because if my analyte sample will bind to this ligand, so it will change the properties of this particular surface, so which will bend the polarized light in some other extent. Previously it was bending in some particular angle, but now that angle might change because there will be change in the conditions of this particular surface from where the polarized light is passing, so due to which there will be change in the refractive index of the medium. So when biomolecules bind to these immobilized ligands on the sensor chip, the refractive index changes, which is detected as a shift in the SPR signal. So in the next slide I'll show you that how the signal changes. But first of all, what things you have to keep in mind, that in this experiment there will be one molecule which is the ligand, which is fixed to the sensor surface. So this is the sensor surface, and I have some fibers through which I have attached this ligand. Now one thing is the binding of the ligand. Now second thing is binding to another molecule or analyte. So the thing which I am analyzing, which sample I am analyzing, that is referred to as analyte, which I have to measure, that whether there will be interaction between my analyte and ligand or not. So the second thing is my analyte, and the response is the third thing which I am recording, so which is recorded in the resonance units, and it is directly proportional to the amount of analyte bound to the surface. So this this response will be directly proportional, because if more and more analyte will bind, then more and more response will be captured. If there will be weak binding or lesser binding, then the response will not change much. So there are some sensograms which we obtain, with which we can identify that what is the strength of the protein protein interactions and how well protein protein interactions are going on. Yeah, so this is one of the example, and this is the BioCore T200 is the device where I am measuring this. So it has 4 flow cells. As you can see, these are the 4 flow cells which are present, which are connected in series. So each experiment we uses the 2 flow cells, either flow cells 1 or 2, or we can use flow cell 3 or 4. So the first is the reference flow cell with no immobilized ligands, whereas the second is the sample flow cell where the, so in this case, in time A and B, this shows 2 flow cells. So first is like a control with which you can compare that if no ligand, if there is no binding of the analyte, so then what kind of peak you obtain, whereas if you apply your sample, then what is the change in that means polarized light angle, that how the light is reflected. So to compare them, we need some as a control and then the sample. So these are 2 flow cells which I am comparing. So now what is happening, that in the second, as I have given the sample, so there will be binding between the sample and the ligand which is attached. So now when plane polarized light will hit on my surface, then its light will bend to some extent. So in this case, here only it is shown, but what is actually happening is that for this flow chamber also the light is passing through here and then it is reflected, but for simplicity they are showing in it on this top surface only. So now what is happening in the case of A, where no sample is given, so light is like at this angle, so you can see that the angle is lesser, but in the case of where the sample is given, the angle increases. Okay, yeah, so the peak is obtained, peak has shifted to the, means angle has increased. Similarly in the resonance signal, signal is showing the rise as soon as the sample is passed. So in that way we can compare that, means how much signal changes. So more and more signal changes, that means stronger interactions are happening. So in that way we can compare different samples and we can go for studying the protein protein interaction strengths and whether protein are interacting or not. So those comparisons can be performed by just looking at the angle of reflection of the light through the medium. So this is all about surface plasmon resonance, and these are the major steps. So immobilization, interaction analysis and regeneration. So this is quite detailed thing, that how the, means how response changes. So this is the baseline response when buffer is there, and when you provide the ligand sample, then also the little bit response is obtained, because buffer has, means in buffer the light is not bending at all, and then by applying the ligand to the sample, there is little bit response obtained. Then still that will be referred to as baseline, because we will obtain the response after the after providing the sample, after giving the analyte, that how well it binds to the ligand. So at the analyte sample you will observe some response, and then again if you stop providing the analyte, you will remove the analyte, then again there will be dissociation phase where there will drop in the response, which will tell you that now no longer interaction is going on, because as more and more analyte will be provided, more and more interaction will happen and more response will be captured. But as you will remove that, there will be dissociation, response will drop, and then ultimately regeneration buffer is given. And this is all about the steps, that what are the 3 different steps in which you analyze the change in the reflective index, and you measure the response and you analyze that how well the response is obtained, so that you can comment that how strong is the interaction between different proteins. Yeah, so now the most awaited technique, the mass spectrometry, is here. So we will go into the detail of this technique because it's well discussed and it is mostly used by people to quantify and identify different proteins. So this is the analytical techniques in which we are analyzing the proteins. Okay, so in this quantification and everything is done on the principle of mass to charge ratios. So in this we compare the mass to charge ratios to identify the peptides, that which peptides are present. So from the spectra we compare them on the basis of their mass to charge and give them some sequence. So there are three main stages of this mass spectrometry technique. First stage is the ionization. So in this, what we do is, firstly we have to ionize those molecules or the particles which we have. So first step is ionization, which includes electrospray ionization or matrix assisted laser desorption ionization. So these are two techniques of ionizing, because ions are the charged molecules, so that's why they'll move in the presence of electric charge, so we have to ionize it. Second is the mass analysis. So after ionization, the particles will have some kind of acceleration through which they will pass. Now the next thing is we have to analyze their mass. So generated ions are separated based on their mass to charge ratios using the mass analyzers, and there are different mass analyzers which are present. One is quadrupole, time of flight, and orbitrap. There are some other as well, just like triple, quadruple, or ion trap. So we will discuss them also in the later slides one by one, that how these different analyzers or ionizers work, and ultimately we have to detect. So in which we will detect the spectra on the basis of how quickly they are obtained. So in the detection, we will detect the separated ions and their abundance is recorded for the quantity purposes if we want to perform quantitation. So resulting data is used to generate a mass spectrum, which will show the relative abundance of ions at a different mass to charge ratio. So on the basis of mass to charge, at different time points we will obtain different ions. So in that way, ultimately we will go for the detection. So this is all about mass spectrometry, means overview of mass spectrometry. Now let's look into one by one, but let's firstly look into this figure. So this is the sample injector port from where we can provide our sample, and then sample will be vaporized, because we will convert it into the form of vapors, so that its ionization could be in a better way. So this electro gun, it will ionize my particles. So I have a protein sample which go inside. Now it is vaporized. Now electron gun will ionize those smaller peptides. So in the samples I have given the peptides. Now these peptides will be ionized with the help of electro gun, so which is the electrospray ionization, so in which ions will be created. Now there are some other accelerators which are present, which will kind of accelerate my movement of my ions, so my ions will move quickly. This magnetic field kind of deflects those ions depending on their mass to charge ratios. There will be some these ions will be deflected in different quantities, so those deflections will help the detector to analyze the different ions which are produced on the basis of their mass to charge ratios. So ultimately detector will identify it. So this is about the mass spectrometry. Now let's look into different steps, step by step procedure. So first thing is the sample extraction. Similarly, we obtain the sample as we have seen previously, that from any tissue or any biological sample we will take the sample. Now we have to digest it, because in mass spectrometry we require the smaller peptides. We do not look the entire protein at the same time, because proteins are quite large, so we need to break it into different peptide units. So for breaking that, we use the triptych digestion methods, or we can also perform chemical digestions, so that we can break the larger protein into smaller peptides. Now next step is the enrichment stage. So in this also we need to make sure that there is certain concentration of peptides which are present. So enrichment stage, and in this we can also go for the IMAC, which is immobilized metal affinity chromatography, phospho, so which will target the phosphorylated proteins, because for example I only want to analyze certain proteins, as we have seen in targeted proteomics. So similarly, if my aim is to just analyze certain proteins, so in enrichment I can also decide that from my sample which proteins I want to analyze. So for that we can go for IMAC, in which we will target the phosphorylated proteins, or we can go for the antibody ubiquitin, in which I will only, in my sample I will only allow the ubiquitinated proteins by using antibodies which are specific to those proteins. So I will only retain those proteins and I will study those proteins only. Or I can also go for the antibody KAC. So KAC is the K is the short form for lysine, so acetylated lysine. So the proteins in which more and more acetylation is present, I can also focus on those proteins by using certain antibodies which are specific to that lysine which is acetylated. So in that way, in enrichment, what I can do is I can increase the concentration of specific molecules by using these techniques, in which certain protein quantities I can increase, I can quantify them and remove other proteins which are not desired to me for analysis. Then the step is fractionation, in which we separate those components, because we have multiple peptides which are present, so we have to convert it into smaller fractions, and this fractionation step is kind of done with the help of liquid chromatography, or I can also go for the centrifugal filtration. So in that way we can have the manageable subsets, because there could be huge peptides which are present, so we create the subsets. So those subsets are kind of created in the form of bins, that this to this range will be kept in this fraction, and then this to this range of peptides will be kept in another fraction. So this is the fractionation step. Then comes cleanup. So in this case, while we are performing digestion, enrichment, fractionation, so there is a possibility that there could be some contaminants or salts. So for that we can go for the cleanup stage, in which we go for the solid phase extraction or protein precipitation, or we can also go for dialysis, in which we will remove the contaminants. Then concentration or dilution. So in this case we adjust the concentration of our sample. For that we can go for evaporation and dilution. Then derivatization, in which I enhance the ionization or detect specific modifications. So means derivatization, you can kind of link it to the labeled methods, in which I can decide first of all that which ions I want to analyze. So this is kind of derivatization, in which we can detect specific modifications. And then ultimately ionization, in which sample ions convert the samples into ions for detection by the electrospray ionization or MALDI, depending on that what analysis I am performing. So I can select any one of those techniques. Then sample loading, in which introduce the prepared sample into the mass spectrometer. So there are some kind of these steps are not in the consecutive manner. There is some disturbance because sample loading is done before ionization. So I will correct it, so don't worry about this. Kind of sample loading, you can ignore this step, otherwise remaining steps are quite remaining steps are clear. But after ionization I won't go for the sample loading, because sample loading is done before the ionization step. So after the ionization, it will ultimately mass analyzers will analyze those ions, and ultimately detector will detect those signals. So those things will happen after this step. Now let's look into each step one by one, that what will happen in the ionization. But before moving further, I'll again ask that if you have any doubts. Is everything clear till now? Okay. So in mass spectrometry, first step is ionization, in which I have to break my peptides into ions. So in ions, there are two types of ions which are produced. One are B type ions and other are Y type ions. So if I study the ions from N terminal, those are referred to as B type ions, and if I am looking at the ions towards my C terminal, then these are the Y type ions. So these N and C terminals are given in perspective of a protein. So protein is made up of amino acids, and each amino acid have an amino terminal end and a C terminal end, in which COOH group is present. So in that way, N to C protein is synthesized from N to C terminal. So in that way, in each peptide it will be having a sequence in the direction N to C. So whenever ions are generated, so from N terminal I'll call them as B type ions, and from Y these will be C terminal, or ions produced from the C terminal. So this is the one step. This is about the ions, how the ions are produced. And in this case I have given the example that why we use B and Y type ions, because they break them at the peptide bond. So whenever in two amino acids they are joined, so they are joined by a bond which is known as peptide bond between the C double bond O and amino terminal end. So this bond is made between the C terminal end of the first amino acid with the N terminal end of the second amino acid. So that is known as peptide bond. So whenever the breakage happening at that point, at that peptide bond is broken, so we are focusing on that peptide bond cleavage, because which will separate my peptide into individual amino acids. So that's why we deal with these B and Y ions and not with the AX or CZ, because CZ ion is kind of breaking my amino terminal end also of the second protein and second amino acid, and it is linking it to first amino acid. So for proper quantification, for obtaining proper ions, we analyze the B and Y ions, because what it does, it is breaking at the peptide bond, which will separate my peptide into individual amino acid units. So with the help of these ions I will be able to identify the amino acid which is present in that peptide. So that's why we analyze these ions. Now there was one paper in which it was very clearly mentioned that how we read these ions. So this paper, you can go through this paper because I have given the link. So in this paper what they have given is, this is the example that this is the stretch of a peptide. So this is peptide, and in this peptide hydrogen bond is, means hydrogen is present towards the N terminal and OH group is present towards the C terminal. So this H specifies actually the N terminal. So NH2 has the extra hydrogen that represent H, and towards the C terminal, as I know that we have the COOH group present towards the C terminal, so this OH is coming from there. Now how we read the ion? So B1 means when I am reading from my N terminal, so H and E. So B1 specifies the first amino acid when I am reading from N terminal. When I say B2, it means I am reading 2 amino acids from my N terminal, so that will be EA. Similarly, B3 will give me mass means mass of the ions, which will be EAE. That means 3 amino acids from the N terminal. So these numbers will tell me number of amino acids which are present, means which individual ions are present. So there are 2 ions, 3, similarly 4 ions are present, 5 ions. So if I want to know the, for example, I have the information of B6 and B7, and I want to know that what is the molecular weight of my 7th amino acid, that is K, which is present in B7. So if I want to know the molecular weight of my lysine, so what I can simply do is I can subtract B7 from B6, which will give me the molecular weight of lysine, that is K, which is present. So I can simply minus 787 from 659, it will give me the molecular weight of lysine. So similarly, in the case of Y ions also, I will move from my C terminal. So from C terminal towards my N terminal I will move. So first Y1 will tell me about K. If I say Y2, it will tell me about E and K. Y3 will tell me about S, E, K. So means what I am doing is I am moving from C terminal towards my N terminal by counting, by increasing the number of amino acid or number of ions at each level, depending on Y3, Y4, depending on the number. So this 4 will specify that 4 amino acids are present. So this is all about their masses. So at each level amino acid are kind of adding. So this is the cumulative mass. So Y2 is providing me mass of both K and E. So similarly, if I want, at any step I want to know the molecular weight, so what I will do, I will subtract the 2 consecutive molecular weight of 2 consecutive ions. So in that way I will be able to know the weight of my previous ion. So in Y1 and Y2, if I subtract, I will be able to know the molecular weight of my E. So if I get the weight of E, so K is already known, which is 147. So for by subtracting 276 from 147, I will be knowing the molecular weight of E. So now there was one question in your assignment in which you were asked that you have to identify that in both there was I think this mass was given for these ions, and you have to identify that whether in which case the 2 amino acids will be same. So firstly what you have to do is you have to identify the consecutive ions, that B6, B7. You cannot subtract B5 from B7 now, because it will not give you the molecular weight of single amino acid. So firstly you have to make sure that ions are consecutive, that is means that are present adjacent to one another, for example 6, 7, and then you have to subtract the mass. So the case where you obtain the similar value after subtracting on both the sides by subtracting, for example y6, y7, you subtract y6, y7 and you subtract b1, b2 and you obtain the similar mass, then it means that both have the same amino acid. So by subtracting you will be able to know the molecular weight of particular amino acid, and if that is nearly same, then you can say that this is same amino acid is present. So both the signals, means both these ions are producing the similar amino acid. So I hope it is clear. Now let's move. Yeah, so similar thing I have shown here also. This is also another paper in which they have taken the example in which there is some modification at this particular amino acid. So there is alpha modification. So what alpha will do, it will add some molecular weight because extra group is present. So we have to take that into consideration, that whenever we will subtract the molecular weight, so in some scenarios where this P is present, we also have to consider this alpha, the molecular weight of alpha also, to identify the molecular weight of the amino acid. So this is about the post-translational modification, that how the molecular weight differs if modification is present. So wherever this P will be present, molecular weight will be added with alpha. So you have to subtract alpha before identifying the molecular weight of the amino acid. Yeah, so now these are three different techniques which are generally used for ionization. One is electrospray ionization. So this is the process that so one thing you have to keep in mind that these all will be converting your sample into different ions. But why we use electrospray ionization? It is generally used when we have the liquid samples, and what it will do is that the solvent sample is firstly dissolved in the solvent, and then there will be a needle from which you will pass the high voltage current. So when high voltage is passed, then that particular sample it will create the charged droplet in the sample and convert it into the ions. And in this case you do it for the liquid samples, and this is a soft ionization method, and it preserves the molecular structure. It do not disturb the structure of the protein or this peptide which you are using in the sample, and it produces multiple charge states. Whereas you use matrix assisted laser desorption ionization. So as the name suggests, matrix assisted, that means there is some matrix, there is some surface to which your sample is attached. Then laser desorption ionization, that means you are using laser light. So if laser irradiates the matrix which cause the ionization of the sample, then it is known as MALDI. And why you use MALDI? You use it for the solid samples. So in this case you have the solid matrix to which your sample is attached, so you use it for the solid samples, and it has better sensitivity. So it depends that what you are analyzing, so depending on that you select any one of these techniques. Then there is another technique which is atmospheric pressure chemical ionization. So this is also for the liquid phase, as we use electrospray ionization, but in this case we use it for the small to medium sized molecules, whereas electrospray ionization is mostly used for the larger biomolecules. So if I want to analyze some big protein, then I will go for electrospray ionization, whereas if I have a smaller pharmaceutical or metabolites which is quite small or medium in size, then I will go for APCI, and it is also similar to that of electrospray ionization, but in this case the sample has to be vaporized, and it is ionized in the presence of a nebulizer gas, and there will be corona discharge. So this is also a kind of a discharge technique in which the current is passed and my sample will be kind of converted to ions in the presence of a nebulizer gas. So this is just one of the difference, and it is effective for the liquid phase samples and good for the compounds which cannot be ionized in the electrospray ionization. So for that we use APCI. Okay, so now one by one I have explained each and every technique. So in electrospray ionization you can go through this if you want to study in more detail that how everything is done in the presence of voltage, how the liquid samples are converted into ions. So for more details you can go through this slide. Similarly for MALDI also I have given stepwise procedure as well as the figure which explains that there is a matrix to which our sample is attached. We pass a laser light which will convert it into smaller ions, and those ions will further flow through the flight tube. So in mass spectrometry this is the ionization stage, then it will pass through the mass analyzer where my ions will be separated on the basis of their mass to charge. So in this case there is time of flight mass analyzer which is used. So in most of the cases in most of the cases MALDI's kind of ionization is further followed by the time of flight mass analyzer. So in that case it will means it will measure the time with which the ions are moving in the flight tube, and ultimately detector will capture that at which speed they are moving. On the basis of that spectrum of signal will be obtained, and ultimately the peptide sequence can be identified. So this is the MALDI, and then I have not gone through APCI because it is similar as the technique which we have discussed earlier, electrospray ionization. So you don't have to go into the detail. Now next step is the mass analysis, in which we use the mass analyzers. So after ionization I have to analyze the mass. So for that we will use the mass analyzers. So there are multiple mass analyzers. Depending on our requirement we can select any one of them. So one is quadrupole. So it uses the oscillating electric field to filter the ions based on their mass to charge ratios. So in this case oscillating electric field is used. It is quite fast and compact, and it is used for the quantitative analysis, and mostly routine analysis and quantitation and monitoring quadrupoles are used. Then comes time of flight, which we have seen previously. So what time of flight is doing, it will measure the time ions take to travel a fixed distance and separating them on the basis of mass to charge ratios. So similar to the previous method only, it will also separate them on the basis of mass to charge, but in addition to that it is also measuring the time which ions are taking, so at what time they are moving, because the ions which will have smaller mass, they will move quite fast as compared to the larger ions. So in this, time is also measured. That's why it is of high resolution, and it can be used for the wide mass range, and it has high sensitivity. Similarly, if we talk about orbitrap, so in this case, as the name suggests, orbitrap, that means it is kind of trapping the ions. So in this case, ions are trapped in an electric field, and their frequency of oscillation is measured to determine their mass to charge ratios, and it is also of high resolution and accuracy, and it is generally used for the complex mixtures. So there are certain ions which will be deflected or which will no longer be trapped. They will move out from that trap, from that orbitrap. So in that way we can capture, the detector will detect that ions. So in that case, complex mixtures, we can go for the orbitrap mass analyzer. Then quadrupole ion trap, this is also the another technique in which ions are trapped in an electric or magnetic field, and they are also analyzed on the basis of their mass to charge ratio, and this allow the multiple stages of mass spectrometry. So means there are some techniques which include multiple things at the same time. So in this case, quadrupole and ion trap both are used at the same time, and then there will be multiple stages, means at one stage the ions will be uh, kind of filtered on the basis of the quadrupole, then at another stage they are filtered on the basis of ion trap. So similarly, there are multiple stages in mass spectrometry in which ions are at different stages, ions are obtained. Then comes triple quadrupole. So in this case, the three quadrupole in series are used to separate the ions. So firstly, one precursor ions will be obtained, then another ions will be obtained. So means at multiple stages ions are filtered, so that is referred to as triple quadrupole. So these are quite sensitive because we are using multiple mass analyzers at each step. That's why these are used for the targeted quantitation and multiple reaction monitoring. So this is all about the mass analyzers. Now I have provided each and every mass analyzer one by one in detail, that how each of them works and what are the steps. So you can go through these slides if you want to know more about them, but we have discussed about them already. Similarly, time of flight, which is kind of measuring the time, and on the basis of mass to charge, molecules are moving. Heavy ions will move slow, they will take more time, whereas lighter ions will move faster, they will take lesser time. Ion detector will detect them. So this is all about the time of flight, and mostly it is used with the MALDI for the solid samples. Then comes orbitrap. So in this case, you can see this is the orbitrap analyzer. So this is a C trap in which what ions will do, ion will continuously rotate or orbit in this trap, and at some time they will escape it, and then analyzer will detect it, and there is amplifier to amplify the signal which is obtained. So ions oscillate around the central electrode, which you can see here in the orbitrap, with their frequency. So there is certain frequency with which if the ions will be of that if ions will be of that particular mass to charge ratio, then they will oscillate in certain frequency, but if the mass to charge ratio varies, then they will escape these oscillations and detector will detect it. So if you have multiple samples, you can go for the orbitrap to compare. Then quadruple ion trap, in which you have two sources of separations. So this is the ring electrodes and ion detector and ion source is present. So in this this is the stable ion oscillation. So in stably, these are the oscillations in which ions are stable and they are oscillating, but this is the discharging of ions with unstable oscillations. So if there will be oscillations will be unstable, then they will discharge and ion detector will be able to detect. So on the basis of mass to charge, separation will be done, because certain mass to charge will oscillate in certain frequency, whereas if other mass to charge will come, then they will not be rotating in that particular frequency and they will escape and can get detected. So in that way, quadruple ion trap will detect that particular ion which have different oscillation pattern or different frequency. Then comes triple quadruple. As we have discussed, there will be three filters, three quadruples in the series, which will at different steps they will filter the ions. So first filtration is mass to charge selection, then comes fragmentation. So after mass to charge ions are obtained, they will further be fragmented with the quadruple collision cell, in which they will collide and then further break into smaller ions. Then in the Q3, the mass to charge selection of fragments will be done. So again, they will be separated on the basis of their mass to charge, and ultimately they will be their signal will be recorded by the detector. So this is about the triple quadruple. Now the last step in mass spectrometry is the detection, where we detect the signals which are obtained. So in this case, ions are collected. Now ion interactions are checked, signals are converted. In certain cases, we have to amplify the signals. Data is obtained and then it is analyzed. So mass spectra is obtained. Now next step is to detect. So what we can detect is, on the basis of the mass to charge, we can compare their relative abundance. We can identify the protein by identifying the peptide sequence and further aligning it to know the protein sequence from where that peptide have been generated. So those things can be done. And then ultimately we have to check for the accuracy. So there are different measures to check that whether the peptide spectrum which we have obtained is accurate or not. So first thing is sensitivity, where we so sensitivity is the means it tells the ability that whether we are able to detect the low concentration of analytes. So we should have the higher sensitivity. So higher sensitivity is better. Then comes resolution. So resolution means we are able to distinguish between the ions which are of closer mass to charge ratios. So if we are able to distinguish that, that is known as resolution. So we require higher resolution. Higher resolution means it's better. Then comes dynamic range. So if we are able to identify the ions at different intensities, so that is known as dynamic range. In the wider dynamic range is mostly favored. There should be, means dynamic range should be wide. Then comes linearity. So relationship between the ion intensity and concentration, it should be linear because it ensures accuracy, because more and more ions are there, then it specifies that concentration of those ions are more. So in that way relationship should be linear. Then comes signal to noise ratio. This is the ratio of the analyte signal to the background noise. So if the signal is better to the noise, then it is better. So there should be higher ratio, that means signal should be high as compared to the noise. And then reproducibility means how consistent your results are. For example, you are using same sample and performing mass spec again and again. So it should be reproducible. You should also obtain the similar results after performing the analysis multiple times. It should not be that each time different results are obtained. So it should be reproducible. That means consistency of result should be obtained under similar conditions. So if better reproducibility is there, that means your results are better. So these are certain measures and this is what we want after analyzing. So if these criteria are fulfilled, which means that the analysis is done correctly and we can rely on that analysis. And then there is so I have given the formula for the mass accuracy, which is in the form of parts per million. So how we check that whether our analysis is correct or not. So we already know the theoretical mass of certain amino acids and there is observed mass. So whenever ions are produced, so they will give some mass. So we will take that observed mass and we will subtract it from the theoretical mass, which we already know that this should be the theoretical mass, because we know the molecular weight of each and every atom which is present in the amino acid, from where we can get the theoretical mass. Now we will subtract my observed mass which we obtained by this mass spec. We will subtract it from the theoretical mass and divide it by the theoretical mass, multiply it with the 10 raised to power 6. That will be referred to as mass accuracy, which means how accurate is my prediction. Then comes resolution. So resolution means in this case we want to distinguish between two different peaks which are obtained. So it is equal to mass divided by FWHM, which is the full width at half maximum. So this distance specifies the FWHM and resolution should be as less as possible, so that if resolution is less, that means how less distance you are able to distinguish. So it should be less. A smaller FWHM indicates higher resolution. So if this FWHM is small, that means our resolution is better, whereas if these are quite large, then means resolution is poor. So these are two things in which we can analyze the accuracy of our analysis. Yeah, then there was another proteomic category which was quantitative proteomics. So I just want to give you the brief hint of the label free quantitation and label based, because you have seen the different tags in the beginning of the slides. So in quantitative proteomics, when you are comparing the quantity of different samples, that protein quantities in different samples, so you go for the label free quantitation and label based quantitation. So label free quantitation, generally it is done where you are comparing between two healthy and diseased individuals. You obtain the extracts, you use trypsin digestion, and then you go for the LCMS, and you are doing these at the different different samples are taken and different experiments are performed, and then comparison is done. Whereas in case of label based, in this case you tag those samples and you are analyzing in the same run. So in similar mass spec only you are doing the analysis, whereas in case of label free you are doing the analysis twice. So in this case there are two healthy individuals and two diseased individuals. Similar thing is obtained, similar peptides are obtained, but now what I am doing is, for each of my sample I am labeling them with some chemical tag. So this chemical tag can have, this tag unit can have multiple means different mass to charge ratio, due to which in the mass spectra which I will obtain in the end, these different samples will produce the spectra with different mass to charge ratio, so I can distinguish between the spectra obtained from each individual sample. So that's why we go for the label based quantitation when we want to analyze multiple samples at the same time. So the relative quantitation and absolute quantitation, so I will distinguish between them. So relative means when you are comparing among two different samples at the same time, and absolute means when you are not comparing it to some control or to some other, but you are identifying for some sample you have and you simply want to identify the quantity of protein present in that sample, so that is known as absolute quantitation. So in case of relative quantitation, what are the labels which can be used? They could be metabolic labels, that could be select. So metabolic labels, simply some organic compounds are used which are labeled, so that is metabolic labeling. Then chemicals can be used, some isotopes can be used, some isobars can be used, so ICAT and ITRQ, these are the chemical labels. Or you can also use the enzymatic labels to label your sample differently and then getting the spectral comparisons. In case of label free, you analyze the peak intensities, spectral counting and spectral TIC. Similarly, for the absolute quantitation also, you can go for iTrack, TMT, tandem mass tags and MRMs, and label free apex. So this is a quite detailed table, but you don't have to go into the detailed flowchart. You just have to take the idea that why you go for the label based analysis. So what are the different labels which can be used? This is all about this slide. Yeah, so now I have given the tools which are generally used for the MS based analysis, because now after mass spectra you have obtained from mass spectrometry, now how you will analyze that, what could be the peptide sequence, how will you identify? So for that you have to use certain tools which will give you the comparisons, which will give you the sequence from the spectra, because spectra are just the mass to charge ratios. You want to know that which is the sequence of this peptide, so for that you have to go through these tools. So these are few of the tools which you can use, and one such tool which was discussed was MASCOT, which is the protein identification search engine for mass spectrometry data. So this we will discuss in detail, that how the MASCOT is used for analyzing the data. So to use this, firstly I will ask that whether everything is clear till now. Okay, thank you. Now we will just quickly look that how we perform the analysis on MASCOT. So to work with MASCOT, firstly you have to log on to their site by using this link. Then this is the front page of the MASCOT, so it appears like this. So you know, these databases or these softwares, they keep on updating them, so this home page keeps on changing. You might not see similar menus after some time. So this is the current home page, how it appears. So this is the arrangement. So now when you will go through this, now what you have to do is, it's showing home, it will give you entire information about the metric science and MASCOT server, that what you can do, what it provide you everything, what are the free services which you can deal, means which you can analyze with it. Now what you have to do is, you have to go to the free trial, because there are no other option to register to this. So you have to go to the free trial, and then it will show you two options. You have to select access MASCOT server, because we want to access the data. So when you will click on the access MASCOT server, then it will open up like this, in which you have to provide your information, your spectral file you can upload. So firstly it will open like this, it will ask you for your name, so you can enter your name and email ID, because this is the first step. So you have to give your email ID so that you can register in this, and they can also send you the results to the email ID. So firstly what I am doing is, I am giving my name and email ID, then I have entered the title, because I want to know, because if I am performing multiple analysis, so I have to distinguish between those analysis. So for that we need to give the title. Now the database we have to select from here. So in this case, I have selected the contaminants as well as SwissProt dataset. So SwissProt is a protein sequence database. So one thing which I want to add, that whenever we are performing the mass spectrometry, then we require a database from where we can match those peptides, because we already should have a sequence of proteins, then only we can identify the peptide sequence which is coming from those proteins. We cannot randomly give any sequence to the spectra. So that's why we require database, then only we can identify that whether it's accurate or not. So SwissProt is one of such protein sequence database. So that's why I have selected protein SwissProt as well as contaminants, so that I can distinguish whether my spectra is coming from protein or contaminant. Then at taxonomy, it depends on you. You can select, for example, if I am only observing the human sample, then I can select the taxonomy, whereas I have kept it default. In case of enzyme, trypsin is kept at default. You can also select other enzymes if you want. If your sample is if the peptides are obtained by some other fragmentation, then you can select at that particular enzyme which you use. Then in quantitation, I am also not changing it. In case of cross linking also, I am not changing it. Then fixed modifications. So if I have attached some, I've done SILAC, I've attached some metabolic agent to my sample, or if there are some modifications, so I can select from this option that what kind of modifications have been done. So for in this case, I've selected carmidomethyl modification, and then at the variable modification, I have selected the oxidation. So it depends that what things you have used while analyzing the mass spectrometry. So in that case, you can select any one of them. So I have selected them because it was mentioned in your assignment. So I am just kind of replicating that same. I am not using the exact same things, but I am just giving you the idea that how things are done. So in that case, rest everything is kept to be by default. We are not changing anything. So now what we will do, we will click on start search. Okay, so before start search, we have to upload the data file. So you can see here, you have to choose a file. So what file you can upload is, so the file which you were given in the folder, that was in the MGF format, which is mascot generic format. So normally the spectra which you obtain from the mass spectrometer is in, it could be in different formats. It could be in XML or any other format. So for these search engines, you have to convert them into their format. So here the desired format is MGF. So you have to convert the spectra into MGF. But in this scenario, you were already given the file with the MGF format. And in discussion, somebody has asked that this file was not opening. So you can open this file in the notepad where you can see. So in that, what is contained in this file is the mass to charge ratios, and different spectral information is collected. So you have to upload the file, you have to start the search. Now the search results will appear in this format. Yeah, so when you are using it for the first time, then it will send you the email to register. You have to verify that email ID and again repeat the same steps, and then you will be able to analyze your data. So in this case, the result will be in this form. It will give you the entire summary. So protein summary will be obtained, which will tell you the significant threshold, which threshold value, that whether the analysis which you have performed is significant or not. So these all things you can look into, which are how many number of non-significant matches are there. You can also look into the non-significant matches, if any. And in this case, it will tell you that to how many targets it has, and how many targets it has identified. So it has identified 6 targets, and this is the FDR. FDR is actually the how many false detections false detection rate is. So what is the false detection? So it is 0%. That means the better it's accurate. So now if you will so in this case, if you will scroll down, then you can see there will be proteins, report builder, and unassigned. So in proteins, for example, I want to know that to which proteins it has the target proteins to which my peptide spectra is matching. So now in those proteins, which protein is matching with greater, which protein is having the better rank, and which has the more score. So that score I can see from these proteins. So there are 6 proteins which are mapping, and then in this case they are showing the score. So my first protein, which was mapping with 187 score, that means which was the best score. So in this case I am selecting that protein. If you will click on this particular arrow, then it will further drop down and it will give you the peptides from that protein which are mapped from, because in peptide spectrum there will be multiple peptides, so a single protein can have multiple peptides. So in this case, firstly it will tell you about the protein which have maximum score, then if you open it, it will also tell you different peptides and what is the score of those peptide mapping. So in this case we can open it by clicking on the arrow, it will drop down. Now it is telling me that there are 7 peptide matches for this particular protein, and it is also telling me the observed and the calculated weight, as well as what is the delta m score, so what is the difference between the score, and then what is expect. So expect is similar, that if lesser is the expect, that means it is not produced by random, it is better. This is telling me the rank, and this is the peptide sequence, that from my spectra what are the peptides are present. So in this way you can compare, you can give your file of spectra and you can get the peptide sequences, you can identify the protein, you can identify the peptides. So this is about the mascot, and there are other things also which you can explore by going to mascot, and in this I have shown it in the wide, that how the peptide sequences are obtained. So in this way you can analyze that from your sample file, means from your mass spectra file, what could be the possible protein and what could be the peptides which are present in your sample. Now we will practice few MCQs. Okay, so I hope till now everything is clear. Okay, so about question number 5 to 8, even I also don't have any clue, but I think the TA, who is the TA with the current supervisor, he will let you know on the discussion forum only, because they will do some changes, I think, because the data, means till now the server has changed a lot, because new data has also been updated. So now maybe that particular file is not aligning to that target. Now target sequences might have changed, due to which the results are also different. So maybe they will give you some bonus scores. They have to give actually, because it's not your fault, so they will update you. I don't have any contact with them, otherwise I would have let you know. Okay, so now let's practice few MCQs which might help you to build up the concepts, and this might help you with your assignment also. So first question is, which of the following is incorrect for mass spectrometry based proteomics? So first statement is, MS based proteomics can help in biomarker discovery. Second is, MS based proteomics can help in screening the mutant peptides. Third is, MS based proteomics can help in protein identification without databases. And fourth is, MS based proteomics can aid in screening of therapeutic targets. So now you have to tell which option is incorrect if in perspective of mass spectrometry. Okay, very good. Yeah, so C is the right option, which tells you that without database you cannot identify the target. So yeah, this is the wrong statement. Otherwise everything can be done with the help of mass spectrometry based proteomics. Now there is another paper which I have come across, which was about the protein identification using MS data. So in this they have shown that, means how the databases are used and how to analyze the spectra. So you can read this paper if you want, but the main takeaway message was that the identity of a peptide is, yeah. So there is one more thing, that when you are using multiple, as we have discussed, that 3 quadrupoles or multiple mass analyzers in series, so what happens is there are different multi-stage MS. So multi-stage means there are multiple stages, one is MS1, one is MS2. So in that scenarios where you have multiple stage mass spectrometry, so the analysis of peptide which you do is at the last stage. So that will be in MS1, MS2, you will analyze the data at the MS2 level, you will compare the identity of peptide at the MS2 level. So keep this point in mind, you will not analyze at MS1 level, you will analyze at MS2 level in case of MS data analysis. And if you want to read more detail, then you can go through this paper. Yeah, then there are 2 techniques which you must be aware of. One is data dependent acquisition and one is data independent acquisition. So many people get confused. They say that data independent acquisition means that you are not using any database, but it is not that. Data dependent acquisition and data independent acquisition are 2 separate things. So in the case of data dependent, what you do is, you at MS1 you perform the survey scan and acquisition is triggered by abundant times. So in data dependent MS, what you do is at MS1, the samples which are obtained in abundant, you are only dealing with those abundant samples, and you will select those abundant samples and you will move forward with the MS2 spectra analysis. Whereas in data independent MS, you will proceed with all those peptides which are produced, irrespective whether they are abundant or at any concentration they are present. So this is the difference between the data dependent and data independent. So in data dependent, this next MS2 will be decided by the ions which are you obtain at the MS1 level on the abundance. So as in this figure you can see that there is some intensity threshold. So if ion is crossing that intensity, then we will select only those peptides and we will study them at the MS2 level, otherwise we will ignore all other. Whereas in case of data independent, irrespective of intensity, I am comparing all these precursor ions for the MS2. So this is the difference between DDA and DIA. It has nothing to do with the database at the peptide identification level. Okay, yeah, so this is one of the difference between them, and in this case it is also showing the popularity. So by the means we already were having the DDA, so by the time it is always popular, but DIA and SWAT has also gained the popularity from 2017 to 2022. So these are also popular, whereas DDA is most popular among all the analyses which are done. So you can go through them. Now next question is, which of the following analyzer is common in both the Orbitrap fusion and triple quadrupole based mass spectrometer? Option A is Orbitrap, option B is quadruple, option C is ion trap, and option D is both Orbitrap and quadruple. So you have to tell that which one mass analyzer is common in both of them. Option C, ion trap. So in triple quadruple, as we discussed, did you see any ion trap mass analyzer there? Okay, so I will not waste much time. So its correct answer is quadruple, because as we have seen in triple quadruple, we have seen there are 3 quadruple in series, whereas in Orbitrap we have 3 different things. So in Orbitrap fusion, what we have is, in Orbitrap fusion we have the quadruple mass filter, we have Orbitrap mass analyzer, and we have ion trap mass analyzer. So in this case what is said is we have to tick the common one. So in both of them the common is quadruple, because in triple quadruple we only have 3 quadruples, we do not have Orbitrap, we do not have ion trap. So the correct answer will be quadruple, and you can go through these figures. This is for the detailed analysis, detailed understanding if you want. Now let's look at the next question. Which of the following chromatography technique separates the protein on the basis of their hydrophobicity? So till now we discussed about liquid chromatography, but these chromatography techniques, there are multiple techniques. So you have to tell that which technique could be used to separate the molecules on the basis of their hydrophobicity. So first is strong cation exchange chromatography, B is gel permeation chromatography, C is reverse phase chromatography, and D is affinity chromatography. So I think we have not discussed it, and I don't think you have discussed it in your lectures also, so I'll just tell you. Okay, nice. Srijita is answering for all the questions. Yeah, so right, the correct answer is reverse phase chromatography. So I have also given a table to give you the idea that for which these these chromatography techniques are used. So these strong cation exchange chromatography is kind of separating on the basis of the charge or ionic interactions. In case of gel permeation, the separation is on the basis of size. In reverse phase chromatography, the separation is on the basis of hydrophobicity. And in case of affinity, as the name suggests, affinity, that means what is the specific binding. So affinity is linked to the binding. So specific binding interactions can be checked with the affinity chromatography. So the correct answer is reverse phase. And in this I have given the representation that how it works. There is a column and the how the separation will happen. So most hydrophobic components, they will elute last, whereas the least hydrophobic will move first. So this is about the technique, how it works. And then next question is, which of the following is correct order of steps in the in-gel digestion protocol? So when you do in-gel digestion, then what are the steps? Dehydration, Comasi destaining, alkylation, trypsin digestion, and reduction. and these are the steps. Okay, so I will tell you the answer only. So the correct answer will be the B. So what are the series of steps? So firstly what you do is you go for Comasi destaining, then you perform dehydration, then reduction step is there. After reduction alkylation is done, and then ultimately trypsin digestion is done, and then after trypsin digestion we will go for the mass spectrometry. So this is about the in-gel digestion protocol, and these are the steps. So this was the question in your previous year assignment, that's why I kept it. So these are the steps. And now the next question is, which of the following group is responsible for an overall same mass in of peptides in an ITRAC reagent, ITRAQ reagent? So one is reporter group, balancer group, hydroxysuccimide group, or peptide group. So I just want to give you quite a little idea about ITRAC, how the labeling is done. So don't worry, I'll quickly end the session, not many questions are left. Okay, so anybody wants to answer? Okay, I will tell you the answer. So the group which is responsible for the overall same mass is the balancer group, and this is the information about all these groups, what is their role. I won't go into the detail, you can go through the slide. Yeah, so the thing which I want to focus on is that how the ITRAC tagging is done. So this is the paper in which they are very clearly assigned. So I will just simply explain it. So in the case of ITRAQ tagging, there are three things. One is the reporter tag, one is the balancer tag, and one is the peptide. So in ITRAQ, which is the isobaric tagging for relative and absolute quantitation, so in that case what you have to do is you have to keep the mass constant, but you will have the reporters which will vary among different samples. So there will be different reporters which will have different molecular weights, but the balancer, the role of balancer is to neutralize you, to kind of balance the weight of two different samples, so that the reporter, because reporter is varying among two samples, so that will influence the molecular weight. So to make sure that in two samples the reporter does not influence their molecular weight, we have to add the balancer. What it will do, it will make the molecular weight addition similar in both the samples in such a way that there will be no influence if you are comparing two samples. So there is no influence on the molecular weight, and the reporters will be different, so you can capture those reporters differently. You will be able to distinguish between two samples. So this is ultimate aim of the balancer, to balance the mass among the two samples, at the same time help you to identify them separately because different molecular weight reporters are attached. So this is about the isobaric tagging and the balancer group. And yeah, this is also one of the representation for your for simplicity, that if your reporter is heavy, then you will use light balance. If your reporter is light, then you have to use the heavy balance, so that both the samples will have similar additional molecular weight brought about to them by the reporter and balance. And there are further more details about ITRAQ and TMT. So TMT came after ITRAC. TMT is the tandem mass tags, and with the ITRAC it was limited to 8 samples, but TMT can also multiplex till 16 samples, so that's why this TMT came up. And in TMT labeling, so this is all about TMT labeling, that how the mass reporter is present, mass normalizer, and amine reactive group. So in isobaric you make sure that the mass is constant, but in this case you don't have to constant the mass. So this is about the TMT. Yeah, so this is all. If you want to go to the detail of any of the techniques, then every link is provided, you can read the papers. And yeah, so this is all about today's session. If you have any doubts, then you can ask. And thank you so much for being patient. Okay, so one thing I want to ask you, Polemy, because you are continuously asking from last two sessions, so are you able to access those presentations which I have given in the previous sessions? Were you able to access them? Because you can access them from the Swayam portal itself. So today also in the evening only I will be uploading these PPTs, as well as the YouTube session also I will upload. So don't worry, I will also give the link to this PPT in that YouTube video description also. So if you don't want to go into the Swayam portal, then you can get the PPT from there. Anything else you want me to provide? because in the last session when we were discussing, so you asked me to provide the YouTube links, so I have added those YouTube links in the end of my slide. If you want to go through the video lectures of those techniques, you can go through them in the previous PPT. So, yeah, okay, so you got them. Okay, yeah, so because 3D visualizations will help you to gain more understanding. Okay, so if there are no further doubts, then I will end this session and we will meet in the next session with the week 4 topics. Okay, thank you so much everyone. Okay, I will end.